So I'm going to go through and kind of show you the lab. So the first thing I'm going to do is show you kind of how the client works and what's in the client. That's kind of the one hurdle that students get over is that, I mean, maybe only 5%, 10% of the students, and then show you what those labs are. So, so what students do is they go to our website and they download a client, and it's Mac or Windows, and we do have Chromebook version, but it doesn't work through the client like this. It's an Android app that gets installed onto Chromebooks from the district level management console. So they work the same way. We have chemistry and a beta version of physics. So when you start the client, so when you install it, it's one click and installs, and then when it first runs, it kind of pulls in some updates and some other information for worksheets. So when you get the client, what it is, it's a little app that runs in between the labs and the cloud, so we can manage all the content and manage the students. So students don't log in, we don't track any of their data. It's pretty standalone type of setting. So when you launch it, you get the Labs tab here, shows you the different labs we have, and they're all an individual product. And so we have chemistry, organic chemistry, biology, physics, and physical science. You notice that they have these levels underneath. That doesn't change the lab. It really only changes the presets, and I'll show you those for the worksheets and the lab activities. So it's like having a room with a door. You go through the door, and then you have this lab with a lab bench, and you can do anything you want on that lab bench. You're not constrained. And so it really is applicable to any level, but what you do is you change the level of the lab activities or the worksheets to match what you're teaching. So we have different content for different levels, and all of that content resides here on this tab. And so you have a subject like chemistry, and you go to chemistry and you say, hey, I'm higher ed, so you'll click higher ed, and then you'll pick, hey, maybe I'm teaching thermodynamics, and then we have all of these PDF files with the different assignments. So let's say that you want them to learn about Hess's law, then you click on that, and here is the content, and it gives you a little bit of background. So we're assuming that most of the background comes from outside, but it does give some context so that students know what they're doing, and then it gives them the step-by-step instructions on what to do. So these are pretty highly structured in the sense that we're giving them instructions, but we follow a pedagogical framework called POGIL, if some of you are familiar with it, Process-Oriented Guided Inquiry Laboratory. So it's about discovering through doing the labs, and it's about following Bloom's taxonomy upward. So we start structured, and then we start doing things a little bit unstructured. So in this experiment, you measure the heats of reaction for 3 different reactions, collect the data, then you go through and do the calculation, and then compare, write down the reactions, and then compare how well the 3 heats add up to give what Hess's law is. And so this is just one example of what it is, and we have about 80 lab activities. So
but you don't have to use the lab activities unless you want to.
Yeah, you don't have to use these. These are just there for you to use, and you choose the ones you want to use. There's no question we have much more content than what you may use, and it's just, in my experience, how we teach chemistry is highly unique from instructor to instructor of what they think is important. So we try to be pretty broad in what we have here. And so this is kind of how the worksheets, they're PDF files. You don't have to have the students access them. You can download them, save them, and host them on your own system if you want. You're welcome to modify them. You're welcome to write your own. So this is the license tab. So there's 2 ways that students get access. You either tell the student to purchase it. So they go to our website and they go purchase for $25 a code, which they put in this box and submit it to activate the product. or schools, either higher ed or K-12 schools, can buy a code that is good for X number of uses, and you tell us X and we sell it to you, and the pricing depends on your size and other things like that. And then that code just gets entered here and gets activated. Another alternative approach, especially when you're starting to get to the 1,000 user level, is we can integrate the code distribution into your LMS through an LTI system. So the students click on a link and we give them the code that way. So it's all about getting this code. And then finally on this client, this is where you specify the level. So biology I have set for higher ed, middle schools, middle or high school, higher ed for physics. And so that's where you specify the level, and I'll show you what that actually does. So I click on chemistry, that's how you get to the lab. And so the lab comes up, and so this is our landing page. So each one of these icons represents a lab bench in the general chemistry lab. Some of these get reused, like quantum mechanics, gases, calorimetry gets reused in physics because it's kind of a cross. It overlaps physics and chemistry instruction. And so this is kind of how it works. You got help, lab book, which I'll show you over here. And over here on these worksheets, these lab activities, this is where we have the corresponding table of contents, if you will, of those same activities. So if I go down here and I say I want them to do the Rutherford backscattering experiment, then what they would do is that they get that PDF of that worksheet, or you hand it to them through your LMS. They would come in here and click on this same link, and the instructions to do that are in the PDF. And then we open the quantum lab and it's preset for that experiment. So we have the ability to preset or rearrange or arrange the lab into a certain configuration depending on what the focus of the activity is. Do we want to focus on the building the experiment? Or do we want them to focus on doing the experiment and focus on analyzing the results or something like that? So in this case, we want to build it for them. And so this is the experiment. So it's a optics table. And then all the labs have these stock rooms. And so here's the stock room for the quantum lab. It's the P-Chemist Delight. It's got lasers and electron guns and light bulbs and these different detectors like spectrometers and video cameras, and these sample holders. So you have oil mist, liquids. This is where the metals would be. And then you can select these different metals and gases. So, so you can mix and match, and you have these modifiers. I'll show you kind of how they work. But you can mix and match and build it on this table and do experiments. And we have all the different permutations and all the different outcomes. And we encourage them to explore. It's really cool for them to find things that don't work, that blow things up or burn or catch on fire. And we have that. So in this case, we have alpha particles, and they're going through a collimator, and they come through and they hit the metal foil. And then we have a detector here that shows the results. So here we can move the gold foil off and then say, hey, this is what I see. I see a small spot of my alpha particles coming through. What happens if I close my shutter? Then I block them. I open my shutter, they come through. So it's just a way for them. So for me, a lot of textbooks that say, here's Rutherford's gold foil experiment. He shot alpha particle at a gold foil and found that the atom was mostly empty space, but they really don't know how he came to that. The students don't know the science or how the science and the logic work to come to that conclusion. And so to them, it's just a fact instead of an opportunity to learn how science works. And so this is a great way to teach them one experiment. And the worksheet for this, we have it for a high school version and a higher ed version. You know, it takes 10, 15 minutes to do this, and then they learn something. And so we have them, so move this detector over to the side and say, hey, I still get alpha particles, but no spot. I can move it over here to the side over here. Looks like nothing's happening, but then you see one over here, and then you can click persist and start saving where these things are hitting. And this is all based on real data. So this, in a sense, really isn't a simulation. It's just using real data and statistics that we gathered when building this. And so this is an example. So you can clear the lab, and then what we have. So here's that whole stockroom again, and so they can mix and match and build these. Each of these bells bring up help. Nobody ever reads this, but it's all here. How the works, how each of the pieces of equipment, and it's always good to read about the simulation and the kind of assumptions and constraints we're working under inside of the simulation. I'm trying to get this thing cold when that bar is covering it. So we also, all the labs also have these clipboards, and the clipboards again have these presets. So you should explore these. Thomson experiment. I mean, here I just click on Millikan, and now we have electrons flying through an oil mist being detected by your video camera, and then you can adjust your electric field. And what you do is you pick a spot and you try to hold it, and then you can time how long they fall and actually measure the charge of the electron. So this lab, I mean, this whole bench, the whole quantum simulation here, it's really geared around experiments that you would never really have your students do because you don't have the equipment or the time or a safety issue. And so here's two-slit diffraction. You can change the wavelength, or you can change the splitting of your two slits, or you can change the how many photons are coming through. You can persist mode is save where they all hit, and then build up the diffraction pattern, talk about Heisenberg's uncertainty principle. So this is just an example of the quantum lab, of what it does. And then each of these benches is the same, all right? So again, quantum is something that not going to reproduce something you're probably doing in the lab already, but it is cool to go use for your lectures and just give it as homework, and give it as group work, have students work together in some way. So here's the inorganic lab. So this is actually the first one. So let me spend some time on this one as well. So this is one that you might reproduce. So a really high fidelity lab replacement. So in this one, I grab a test tube, I put it in the stand, and I see a real picture of what's in it, which is nothing. So I can add these reagents, any of these cations, any order, any combination I want, and I'm going to see a real picture of what's there. So now that I have something in here, these lab manipulations work. So I can click on the flame test and show a flame test of that mixture. So you see the green for the copper. I can do a flame test with a cobalt filter. So we can do flame tests of any combination, anything that you want here. We can decant, we can divide a test tube, we can turn on heat, see what happens to the chemistry, pH of the solution, pH of the vapor, smell it, and so on. So lots of things. So now you have these 11 reagents that you can add any order, any combination, any number of times. So I can go to pH 4, which is, and see, hey, there's my litmus paper, pH 4, here's pH 7, here's pH 10, and I show the chemistry of what's going on when you go to pH 10. So everything forms a precipitate, and I could centrifuge that if I want and force it all to the bottom, or I can go to sodium hydroxide and then show that the chromium is in solution, but I add sodium. So if I do a flame test now, I'm not going to be able to see the copper because it gets masked by the sodium flame. So I can centrifuge that, I can decant, and so in decanting, I'm left with my two mixtures here. Now I can add ammonia, and copper forms ammonia, but if you go to pH 10, they're both amine complexes. Go to NaOH, only one is. Centrifuge that, decant, and now I've done my separation. You can click unknowns here. You can click clipboard to give them different kinds of unknowns that are assigned. These are practice unknowns. We also have here a lab book for them to take notes, and they can save different things. Like right here, I just saved my inorganic image that they can put in their notes. And these lab books can be saved and delivered to you, emailed or uploaded to your LMS. So lots of things that you can do. And so this is classic. This is just a classic inorganic, and you can do any level you want with it. So any questions before I kind of go on?
Feel free to just unmute your line if you want to directly ask a question. There have been several questions on the lab notebooks. If it helps, Brian, I created a lab notebook that would show high school labs often spend so much time teaching students how to write out all the procedures. So if you actually have your students within that lab notebook, Brian, if you open your lab book there, you can have students write pages and pages of their procedures and all of the things they're observing, and then they can just send it to you, and you can actually do your lab notebook replacement within this, where they're analyzing all of the data, whether they find it in the bio lab or the physics lab. Any of it can be saved in this notebook and then shared.
So all the results, so like all those detectors that I showed inside of the quantum lab can all be saved. The way the lab book works is it's divided into sections. So you can say this is experiment 1 or whatever you want to make it. And then you can say, I need to take good notes. And so you can save this. And then what you can do is you can create a new section and say, have them. So this is exactly what I'm doing is, I was teaching some schools call it the honors. It's our majors lab class this semester for BYU. And we were getting into the qual. So we're just using the inorganic lab for the rest of the semester. And instead of having them keep all their notes in a real notebook that they take a picture and then upload, I'm just having them keep their notes in this. And I'm having them upload that with their answers to their unknowns that I give them, that I hand out in the inorganic lab. So let me show you that a little bit more about how unknowns work. I can come over here and select cations, and then I can define, or you tell them how to define how the random optimization is going to be. So I'm going to say I can get 0, which is water, or a maximum of 3, and I can create this unknown. And then here's an unknown, and voila, I already know the answer. It's copper. It's cobalt just because of the colors, but you can have them go to sodium hydroxide, centrifuge it, you decant, and now you're left with that. Go to ammonia, and so you get that. So I can go to pH. I can go kind of confirm it by adding carbonate and kind of go to pH 10 here, add some carbonate, and it doesn't work because I have the amine complex, pH 7. So I know it's copper. Then I come to my notebook and I get this little report button. So now I have this extra button up here, and I click report, and on the report button, I just say click cobalt and click submit, and then it gets saved here in the notebook, and then they can practice all they want. And then what you can do is, and then over here, you can get these basic unknowns like the chlorides, for example. Let's clear this and let's get alkaline metal unknown. And here it is, and it's unknown 5. And so we give you a key of what is in unknown 5, and you grade that. Another option is to, come down here and you click your unknowns here. So here's like group 2 sulfates. And this one is randomized, so they don't know what number it is. And they come over here to the lab book and they click report. And then you would just grade based on what their answer is inside their notebook. So they would click here and say, I got magnesium, click submit, I got 2 wrong. You would just upload this, have your TA or you go through and read these and grade them. All right, so that's kind of how it works. So I kind of want to show biology too, but you know, gases, let me just kind of show you the labs, but not really do anything too much in them. So in the gases lab, the whole idea is you have an ideal gas and you have real gases. So these are the most accurate equations of state for these gases, like steam tables for the water and the carbon dioxide. So these aren't just van der Waals models. We actually have a van der Waals gas that you can pick. And then what you do is you have these different experiments. So here's one with a balloon. And I can bring this out. And so out here, then you pick one of these gases and fill it up with that gas, fill the balloon up, and then you can zoom in and do these experiments, change your units. And then what you do is you save this data to a lab book, for example. And, you know, here, what happens if I increase my pressure? What happens to the volume? So you stop saving, click on the lab book, and then here's all that data that they can then analyze and plot it in certain ways. So we have 4 different experiments you can do there. We have the classic titrations experiment. So you have beakers, pipettes, you know, pH meters, conductivity meters, make them calibrated. We have graduated cylinders, analytical balances, and then we have all of these different acids and bases and oxidants, reductants, and you can titrate any acid with any base. So if you're wondering how we do the pH calculations, we use a mass balance, charge balance approach without any assumptions. And we apply activity coefficients as well as volumetric correction. So we use partial molar volumes, partial molar heat capacities to actually correct the volume addition and concentration levels, and Debye-Huckel, extended Debye-Huckel to calculate activity coefficients. So they can do any titration. We also have presets here. So here's a preset. So you can see here's the meniscus, pH meter, conductivity meter, a graph, and we also have these 8 different indicators that they can use. So this is an unknown acetic acid. So you can see the the graph is here. You can save this data to the lab book, and so on. So they can also create unknowns here on this thing as well. Create an unknown. So how that one works, calorimetry. So this is really nice. So, you know, possibly, so we know that Western Governors University, they use these labs for their PChem course, their online PChem course, as well as Gen Chem, because you can do some pretty advanced calorimetry here. So we have bomb calorimeters, Dewar and a coffee cup. We have, you know, not just a metal, but you have all of these different metals for which you can measure the heat capacity. Students like to go find which metals react, like cesium with the water, and blow things up. So we have all of these heat capacities. And then you have organics and reactions and salts, and so you can do these. Here's preset, for example, of, you know, let's say that they want to measure the heat capacity of sodium nitrate. actually, let's go do another one that's even better, is do what's the ice packs. What they use for ice packs is ammonium nitrate. So here's ammonium nitrate. So this is preset. We got a thermometer, we have a graph. You can click save the data, and we've already got the mass. It's already measured out in this preset, but you can make them do it. So it's 2 grams, and you drag that over, you add that, and now you see here in the graph that it's cooling. You can advance your time, and so we have a great worksheet that talks about enthalpy versus entropy. But for a PChem, you wouldn't use the preset. You would just say, hey, why don't you build this and design this experiment for how you would do Hess's law. And it's not just these bottles. We have like 100 different. We got 100 different bottles, I think, for measuring combustion calorimetry and heats of formation and things like that. So that's chemistry, general chemistry. So you can see that the level can be anywhere. It's, it's not too hard for high school. I mean, it's really how you write the worksheets and the level, and it's really great for high school and high school there. So any questions?
There have been a couple specific questions from Ruth Ann about, like, could we determine the acid in Sprite or Coke, which I think is a great idea. Just don't have that yet.
Determine the what?
How much acid is in Sprite, for example.
Yeah, we didn't, I mean, I don't think anybody has the time, given what the constraints we're working under, to go online. But technically, we could add that as a bottle in our stockroom. If you really wanted to do that, I could show you how to mess with our database to define the chemicals and stuff. So it's technically possible to do that. We don't have to change any code, but then, you would have to have your own delivery for your own database and own custom version of that. And if that's something you want to tackle, we'd certainly teach you how to do it. But right now, it's not included in the default. But, I mean, if it was me and I was too lazy to go through all that work, right? Because I don't have the time, what I would do is I'd come over here to my unknown. And what I would do is I would go, let's say that you wanted to analyze vinegar, for example. What I would do is come down here to, where is our acetic acid, right? So I would pick acetic acid as an unknown, and I would then define the concentration in vinegar that I want, so what kind of vinegar. And then I don't know what that is in terms of molarity, but then then what you can do is you can change your, your constraints here to be like 0.07 or something like that. And then what you do is you then create a vinegar unknown, and it's going to be somewhere in there, and have them report that. Or you can just have it all the same, right? Move your constraints down so you don't have any variability, and it gives everybody the same unknown. So that would be a way of doing vinegar. What's in Sprite? What's the acid in? Is it citric acid? I don't think we have citric acid. So anyway, that's an idea of how you could do it. All right. Anything else, Heather?
Let's go ahead into biology.
All right. Biology. So I'm not a biologist. So I had 2 colleagues at BYU who were co-authors on this with me. One's an entomologist and the other one is an evolutionary biologist. So the idea on this one was, when we started thinking about doing biology, each discipline is pretty unique in the experiments and what's important to them. So biology, a lot of it is field-based, and a lot of biology has lots of these, what I would call smaller, more targeted experiments. And so how do you capture all of those different experiments? And so the approach that we took, based on the experts who are really leaders in their field, is modern biology is all about diversity. So it's all about how do we relate, how do we compare one species to another and organize them to know their relationships, to know how things evolved over time. So it's all about diversity and evolution. So what we did is we created these 5 different lab benches that explore diversity from 4 different levels and then organize them in a systematics lab. So in this one, microscopy would use microscopes. How do we look at species from the outside? How do we look at them when we do SEM or TEM? We do genetics experiments, molecular biology experiments, ecology. And then based on that data, how do we organize our species? So in the microscopy lab, again, there's almost an unlimited amount of staining you could do. So our focus here was, what information does each of the microscopes give us, and what is the right microscope for the right situation? And so what we have in each of these labs, is a species selector, and we've created a virtual world with about 175 species. And so what you can do is pick these species, and in each of these labs, so we have different subsets for the different labs, and we show a picture here. We can show you some basic information on it. And you can use the species selector to organize and say, okay, where's all my plants? Where's onions? And we're going to show them these thumbnails of these different species, looking at them with different microscopes, so they learn, what they're going to see. So you have protista, you have alphabetical, and then you can go over here to characters and look at all of the invertebrate, or you can go here to classification and look at all the fish, and so biomes and bimodals. And so all of them is about organizing and looking. So if I go down here to paramecium, this is my favorite one to show. We're going to show a dissecting scope and an SEM. So here's a video that we took. So we took almost all of these pictures we took. So here's a video that they can play and say, okay, this is what I see with a visible light microscope for a compound scope. Here's a different magnification. Here's a different magnification. And then we have these with different stains, so they can see different, and then identify different parts as they go through. And then they come over here to the SEM, and we can show them what they see on a paramecium with that kind of microscope and see what kind of resolution they can see. So to me, as a non-biologist, I just think this is so amazingly cool to see this kind of resolution and then see which tool is used for the right thing. So we have worksheets that use this to talk about microscopy and how to use it and the different pieces of information. So we have a database of about 3,500 images that they use. So this is our high school version. So all of our worksheets that we have for this are organized by NGSS.
Brian, could you go to molecular or to evolution first before genetics? Sorry.
Sure. I got to move this out. I don't know if you guys can see those things. I got to move them out of the way to get to stuff. So here's molecular. So in the molecular lab, the idea here is to pick a species, pick a primer, amplify a certain gene using PCR, verify that with a gel, and then sequence it with the sequencer and look at the sequence and then analyze the sequence about what we know about how species are related at the DNA level. All right, so what we did for each of our species in our database, what we did was we went to GenBank and we divided the species into groups based on their characters or whatever, like plants or mammals, and then different kinds of insects. And we identified a set of genes, sometimes 20, sometimes little as 20 genes, up to maybe 60 genes for mammals. And we created a mini genome for these species based on real gene sequences of a particular set of genes. And then we added random DNA code in between these, and we created these little mini genomes. So what you can do is you can pick, like for barnacle, and you can grab an epinorth tube and put it over here. You can click over here and label the epinorth tube if you wanted. And so you can click your pipette, add it. So now I've just added a DNA. I can go add a tagged nucleotide, phosphorytically tagged, and then I can use TAC. And so I've added my, what I need for amplification. And then what we do is, based on the species they've picked, we then put in the primers they need for the genes that we have for that species. And then we throw in some distractor, and then you can do that. And then you can always define your own. So maybe you want to teach them what happens if your primer isn't long enough. Can you amplify multiple genes and get a mixture? And so we allow for all of that. And so you can pick the right primer. Now I have no idea what the right primer is. So I'm going to use my preset here, and I'm going to pick a preset we have for a fruit fly and a house fly, and all the primers have been added. So I'm going to go add that to my PCR and close my lid, turn on my controls, turn on my animation, and I'm going to click play, and this guy's going to start doing the PCR. So now it's in a warming step. So you could sit here and watch it for 3 hours if you wanted, and it's going to be a real-time simulation. So we're warming, trying to get up to temperature, but I have a timer here that I can advance. So I just went up a minute, and I'm in the denature stage of things, and so it's going up, and I can now advance to my anneal. I can advance again to go. Now I'm going to elongation, so I kind of missed it because I'm only advancing by 1 minute. So here's my animation showing elongation. So we have these animations showing what's going on, and then we start showing that we're amplifying these things as they go. So
Brian, could you show one of our forensics types worksheets? Right, because this is kind of overwhelming to a high school student, how much is here. Could you show how they could do one of these, the forensics experiments that we have?
Sure.
And how much direction they would get.
Yeah, good idea. So obviously these labs can be super advanced. So like I said, though, we have presets. So here's can I just kind of let me complete the process here a little bit. No, you're good. I just want to, that's a good thing to do. But I just kind of want to show you how, you can come here and do the gel here and turn on your power supply, and then you can control things, but I'm just going to advance my time and do a gel and prove that, that I actually amplified, and then you can use this thing to measure what their mass is and say, hey, I actually got 2 spots. And then what you could do is, let's go turn this guy off first. And then what I can do is actually go over here and sequence it. Now, and click start and start through this sequencing process. Now, so here's, for example, I just advanced a minute, so I'm sequencing. So the idea, though, is using these presets is we really use these, like here's an unknown shark fin sample. Here's DNA from a mammal bone and tooth samples, and here's DNA and other things. So these worksheets we have for biology is, that's the wrong one. So worksheets, and we'll go back to biology, high school, and natural. Is this it? Here it is, shark fin sequencing, for example, right? So, so in this virtual lab activity, you will act as a wildlife forensic specialist. You will use DNA sequencing to determine if the great white shark is being illegally hunted and traded. So here's the case, then here's the procedure. So we step them through each of these little steps, show them pictures of how to do it, add the PCR reagent. So we tell them, you know, that we're going to be targeting the 12S gene as a sequence. We tell them exactly what primers they should add. So here's a left primer, here's a right primer. We add those two, then you run it into the sequencer, and then you save each of these sequences that they do, and then so it's organize them, and then we have them do these conclusions. So then we have their questions, you know, and have them go through it. So this is one example of a forensic one. We have a tiger DNA one, DNA profiling, fish cousins. So lots of different things that you can do on what you do. So again, it's the lab is the lab. It's what you do with it as the instructor. So we went through and wrote some. So our idea was we wrote some content. We developed content that could be drop-in, and you really don't have to do anything except, here, go do this worksheet. Here's the instructions. Students go do it. And that's generally how it works, and it works really well. But you can modify it. You can add your own, you know, content, and we're certainly welcome to do anything you want and modify what we've provided for you.
Can you show genetics?
Yep. So in the genetics lab, so in genetics, again, we have a smaller set of species here for which we created these genetic models. So for example, we could do Mendelian's pea plant. And what we have is we defined for these different traits, you can pick a trait, and we have the correct statistical genetic model for each trait for each of these species up here. So the genetic model could be simple Mendelian, you could have linkage, you can have epistasis, you can have incomplete dominance, and so on. And I didn't even know all of them. We have all of these different genetic models. So the students don't know what genetic model is. All they know is they're going to pick one or two. So you can pick multiple traits, for example, and then you can randomize the parent and sit here and randomize the parent for here. And so here's a homozygote, heterozygote, and then what they can do is cross these pea plants. So we cross them, and you can keep on crossing these, so you can do multiple examples of it. And then you can get your species. So we generally, you know, use the number of crosses, gives you the number of offspring that is typical for those kind of crosses. And so what you can do is start comparing, you know, hey, how come this is 19 and this is 21? Well, because it's statistics, right? And so here's tall inflated, tall constricted, short inflated, short constricted. And what you can do is then mouse over each of the individual offspring and see what the actual phenotype was for each of these different kinds of pea plants. And so they can do a pea plant experiment. They can go here to fruit fly and say, you know, these weird things like, what is it, leg-headed, like here, make it homozygote and do a cross, and boom, they're all leg-headed and look at a leg sticking out of a head. Or, they can reset this and pick multiple things, and you can do human, and do like blood type. So you can do, 3A, B, and O, and then with Bombay type and go do that. Or you can do what most people think as blood type is, it's just simple A, B, and O, even though it's not right. And color blindness, do sex-linked genes. And so if they're a normal carrier and a male is normal, what would happen? Cross. So, you start seeing how many male colorblind you get, and the females are always normal. So you can do sex-linked genes and do dogs and do incomplete dominance and stuff like that. So lots of things. That's how you would do it. If you really want to get advanced, we have population genetics, where you can go track gene frequency over time. So here is like just statistical random drift over time. And then you can say, hey, what happens if my gene is mutating, at only 0.1%, right? Or 0.019%. And you can see how your frequencies migrate. And you can do fitness. What happens if there's different types of fitness parameters? All of this can be saved to your lab book. So any questions about that one?
So Shauna just asked about if we've linked into the AP Bio curriculum or the NGSS standards. So what I did was I went through all the NGSS standards and the AP Bio standards and made a big, huge correlation table of which of our worksheets, the curriculum that we currently have, matches up with each of the standards within each of those curricula. So that's available on the website. It's there, or you're welcome to email us at any point with any questions about this. We are happy to respond.
So this is the Ecology lab, and as far as I know, because I looked through all the literature, we are the only robust ecological, even on a research basis, ecology simulation, a phenomenological simulation of what happens when you put species into certain biomes and let them interact with each other. So what you can do is you can define your abiotic variables. So you can pick, a marine biome, for example, or you can pick tropical forest biome, and you can pick these different biomes, pick their latitudes, elevations, temperature, rainy seasons, define nutrient levels, and all these different things. And then what you can do is pick species and throw these species into these biomes, and then see what happens to them. Most of the time they just die because they're wrong species in the wrong biome. But you can also add population, so you can have migration, you can have a catastrophe where you like remove a species, and how does the system respond to that? So lots of things like that you can do. But again, we have these presets. So let me show you a classic wolf and elk. So in this preset, I'm in a temperate deciduous forest, and I have elk and wolf. My initial populations are 2,000 elk and 100 wolf. So what happens? So I click time, and so the simulation starts, and I'm tracking over here the, I don't know if you guys, can you see this little window I'm moving around? So
No.
You can see the graphs. So then we show the population. So you can have up to 6 species in the simulation, and they're just interacting. So what we do is we assume the elk has, so like here it says excess food. We assume the elk has all the food it needs. So it's not going to be limited by food. It's just going to grow. But then the wolf population, as you can tell, is going up, but the elk is going down because there's no controls on the elk on that eating. So the elk population goes down, it hits a minimum, and then the wolf will then crash because they don't have enough food. So they start dying off, and then they hit a minimum there. And so now the elk start going up. and then the wolf, and then you're just going to start kind of repeating this pattern. Is when I created this this algorithm, I thought it was wrong because I was taught that you reach an equilibrium, a steady state, but you don't. You always see oscillations, and so you build up these kind of, so you reach these oscillations, and then they can start all kinds of different things and see, can you actually kill them off? And it's actually pretty hard to kill them off if it's just for food. If you kill just for sport, like with people, then it's pretty easy to kill off a certain species. So so this is a way for them to learn ecology and balancing balancing the needs of this. And then the last one here is Systematics Lab, where the whole idea here is you have what I call a keep and discard pile of different species, and then you can pick different phylogenetic organizations. So simple Linnaean, a simple made up one to show how it works, but why you have to be more detailed. So this is one where, for example, what if I want to decide where does an elk fit, right? This is totally, that doesn't work, but you would say, oh, it's multicellular and it's mobile and it's got legs, right? It's wingless, okay? And it's got shellless and no external. It's got 4 limbs. So it's quadruped. And so it's interesting to see, so you can see pictures or you can see text. So you can see that, yeah, these are all related to each other, but not everything. A salamander you wouldn't consider to be very related. So and so it's antlered, and so we get elk. So this is a way, but it's wrong. It just shows them how organizing works. So it's a really simple one. And then we have these like the real honest to goodness tree of life, the current state of the art phylogenetic tree. And then we can come down here to more simple ones that kind of work the way they should. So 3 domains, for example, and then you go through and so we have worksheets that step them through the process. And the kind of the way it works is you mouse over one of these guys. So we see what the traits are for eukarya. And then if they don't know what a cytoskeleton is, they can click on that and read about it in the glossary. And so these are the traits they're looking for. So it's not just like they guess, is they start figuring out what a nucleus is and how to use that to go, to go do it. So as soon as I go to eukarya, these guys get discarded. So it's a game to get students to learn how to organize and why it's so important that we do the organization. As a chemist, I didn't really get this until my colleague said, well, it's just like the periodic table. Chemistry really didn't take off until we organized the elements, till Mendeleev organized the elements. So organizing the species is how we do it. So do you guys want me to do physics? Any other questions?
Why don't we save physics for our 5 p.m. session, Brian?
Okay. Sounds great. Any other questions for you?
Okay, Ruth Ann says she's good. Shauna, hopefully we've answered most of your questions. She says she's good too.
Great. That's good. So hopefully this has been helpful. And I think the key is to realize that, I mean, the metaphor I use is we have a lab at BYU that we use for like our non-majors class, but we also use it for our majors. We also use it for a graduate class for inorganic chemistry. And we also use it for like elementary students who come in for tours to do experiments. So the room itself can do anything because we have all the equipment, but we as the teachers decide what equipment to use and at what level. And so that's the metaphor that we try to use for our lab benches. It's just because they're complicated and they can do a lot doesn't mean you do a lot. And so that's why we try to have the content match the level and be useful for those different students. So let us know, contact us. You got the, you know, you can have the community site, or you have our webpage, and you email, and all those emails get to Heather or I, and we'll be glad to answer your questions. All right. I'm going to assume you're all okay and you got things to do. So thanks for your time. Oops, you're muted, Heather.
Thank you all.
Bye.